There are many organisms which you carry, but they become pathogens. You carry many yeasts in your gut, but when you get sick, admitted to hospital, and you are seriously sick, the same organisms circulate in your blood and they are killers. My job involves clinical diagnostic work, teaching the postgraduates and undergraduates who are in the medical school, and also a bit of research. We can synthesize many things from the microbes. Even there is research to synthesize some petrol. They produce these antibiotics for their own protection, but we isolate them and we use them for our treatment. I think microbiology is God.
Welcome to Daktari Tone, where voices, perspectives, and sounds relating to healthcare practitioners and matters health are amplified. This is a podcast by Daktari Online, created for healthcare professionals across Kenya. Each episode brings you honest conversations, expert insights, and real-life stories from the front lines of care. Hello and welcome to the Daktari Tone podcast, where voices, sounds, and perspectives of healthcare practitioners are amplified. Today, we're going to delve into a world that exists around us, but it's unseen. The world of microbes, infections, leadership, health. And I'm joined with Professor Gunturu Revathi. I'm a microbiologist and a specialist in infectious diseases. Thank you so much for having us, Professor Gunturu. I'm going to give you a chance to introduce yourself and let us, get to know a little bit about your world.
Thank you very much. Thank you very much for this interaction. I'm Gunturu Revathi. I'm head of clinical microbiology, the diagnostic section of microbiology within the Department of Pathology at the Aga Khan University Hospital. I'm a full-time faculty of the university. My job involves clinical diagnostic work, teaching the postgraduates and undergraduates who are in the medical school, and also a bit of research. That's, I think, about my profession. I trained as a microbiologist, a monospecialist, which is you don't do any other specialties, you work only within your specialty. And in Kenya particularly, I have worked only in the area of microbiology. And I have supervised many postgraduate thesis projects, taught lots of students, many programs, and I worked 10 years in Kenyatta National Hospital and the Associated College of Health Sciences, University of Nairobi, as a full faculty before I joined Aga Khan University. And I came here to establish the postgraduate programs as a full-time faculty. And I started building the microbiology lab from the scratch. And I've spent almost 19 to 20 years in Aga Khan now. That's my description.
That is almost 30 years of being in the lab or in class talking about biology, microbiology.
If I count from my training days, postgraduate, when I joined as a postgraduate in All India Institute of Medical Sciences in New Delhi, all the postgraduates are teaching the undergraduates. So I started teaching when I was a student myself. So I used to lecture, I used to run practicals and vivas, examinations, and all that I did when I was training as a postgraduate myself. So if you count all those years, maybe it's much more than 30
years. But it's over 30 years. Oof. I would like to know how that has been like. And also the fact that now, you've also brought new generations of other microbiologists like you. Anyway, so we'd like to meet the person behind the microscope, the human side, and also the science side of, Professor Gunturu. And so to start me off, the first question I have for you is, what pulled you towards microbiology? Was it planned? Was it a life? Or like, did life surprise you?
It wasn't a surprise. It was a plan which gradually developed. I don't say that it was sudden. It was a sudden decision or anything. I developed interest in microbiology during my undergraduate days. when I was in Usmania University in the medical college. We start microbiology in the third year, and basically it was the teachers who were fantastic. Professor Radha Bhaskar was a fantastic teacher. She inculcated very deep curiosity about microbiology in us. And we had Professor Bhaskaran who taught us. He used to take personal interest in each student. It's fantastic. We are unable to do it now, but we were small batches. We were just 36 students in the class. So they basically ignited the curiosity in us in microbiology. Usually microbiology is not considered a very interesting subject or remunerative subject. People pay more attention to surgery and other things which are clinically oriented. You go into private practice and either you earn money or you become famous. Those are the objectives of the students. But they inspired us to go and learn more about microbiology. And also at that time, we had a new institute which was set up in Hyderabad, which is my hometown. My mother lives there now. After long years all over the globe, she settled down there now. So it was Institute of Preventive Medicine, and hepatitis B research projects were running there. And hepatitis B at that time was a very glamorous subject because there wasn't much knowledge. There was no vaccine. Not much knowledge about the virus was available. It was called Australia antigen. It was not even called hepatitis B virus. It was called Australia antigen. So those days, this institute also inspired us into research. So I developed this interest in microbiology at that time. After graduation, when the postgraduate choices came, this was my first choice because of the teachers in the department and what the department was doing. The department was already into research. There was a research project in malaria which was going on, which was funded by Indian Council of Medical Research at that time. This was long ago, 35, 36 years ago. And there was also a lot of other research projects which were going on, and we had these exciting teachers. So it was my choice to go into post-graduation in that department.
Okay, and with all that, I'm also seeing you being in this, line of being a microbiologist, and the various, because you've even mentioned when hepatitis was called Australia antigen, and so it's giving the fact that you've seen the research evolve, getting solutions to things that even didn't have vaccines. You being part of, I believe, even innovations and more a solution based to microbiology. Forgive me, but it's not my area, so. But yeah, so okay. When I hear the term microbiology, it reminds me of my biology class when I'm in. We used to have experiment sessions, and we'd go to the lab. Biology wasn't, out to my favorite subject. Okay, I did well, but then it had a lot of rules. It had a lot of Latin words and all that, and you had to get all that right. And then you get there, you have to use the lab, use a lot of microscopes and all that. It was interesting, of course, but now getting to get the answer, oof. But I believe that was the whole process of even us getting now to have, separate microbiologists and journalists. So no, on that, many of us will just tend to think that microbiology is, just the lab and petri dishes, and the fact that you've also said that most people in the medical space think that it's not really that interesting or fun, unlike other spaces like surgery and all that. But then if maybe you could tell us the most, according to Professor Gunturu, what's that interesting,
I'm a bit surprised the way you described microbiology. That's not how everybody looks at it. I'm a bit surprised because people find to read in the newspapers about COVID very exciting. They don't find that this is something dull and drab, or about TB or malaria.
You got me wrong. Reading about it, and even because I have written about some of that, obviously. And in fact, AMR is at the moment, because it's also very rampant, it's an exciting topic that I'll get into, because most of us even do the most wrong.
That also surprises me.
We will get there. I know.
AMR is exciting.
Yes, it is very exciting. But then, I'm talking about now the fact that you, as now the specialist behind the lab, us telling stories and even talking about it, we're going to talk about just the like the icing on the cake, but then the depth part of the mixing is done by you people, which honestly, for those who are interested, they'll get it to be fun. But then for some, like myself, just from back in school, some things were scary to even understand, and
that's very sad. It speaks for the teachers and the program.
Yes.
So I think the schools also have a lot to do with this, to have some wrong impressions about a particular subject like biology. Biology is basis of life. Biology is all about life. So if a student found it very dull and drab and difficult and something you need to mug up, that's very sad. But it's not always like that. And the way you described, that is not how a diagnostic microbiology goes. Microbiology is microbiology, but it has many branches, special branches. There is agricultural microbiology, which talks about all plant pathogens. And it's very important. Agriculture is what gives us food. So if we don't protect our crops, we have no food. So it has great economic impact. Soil microbiology, which is about soil, and it directly connects to our food, animals and other crops and fruits and vegetables and everything. Then you have marine microbiology, which is all about the ocean and the microbes in the ocean, which are in the water, in the depth of the ocean, and then in the animals which are living in the ocean. Most of them are also food animals, a lot of them. So it concerns, though, that is a special microbiology. Now, there are many branches. Human pathology and the microbiology related to human disease is a special branch. Then microbiology related to animal diseases is another branch. Then microbiology related to our environment is a special branch. Currently, I don't say it's a new concept. It has been around for 10 years and WHO propagated it. One health concept. That is, the environmental, human, animal, agricultural, plant, health, everything is interconnected, and it is also connected to human body, animal body, all the microbes which are living inside this animal body, also outside the animal body. So we have many microorganisms which are our normal flora on the skin, in our mouth, in our eyes, all over our body, on the surface. Inside our body, in all our internal organs, there is normal flora. And in the recent past 10 years, this knowledge has come out of Human Microbiome Project, which was a multi-billion-dollar project which was funded by the US administration at that time, because we had very visionary presidents who supported this research, and only US could do it. They had the resources for it, rich enough to fund those machines, those reagents, and the people behind it. Of course, the brains behind the Human Microbiome Project were from all over the globe, China, India, Africa, everywhere. And that project told us that our health is dictated by our microbiome, including our immunity, our resistance to disease, our normal health in the normal conditions, what is called homeostasis. Homeostasis is maintaining a balance of good health, is all due to the microbiome. And if the microbiome is disturbed by many things, environmental or food or drugs or medicines, for example, antibiotics can kill the microbiome, alter the microbiome. it has very, very far-reaching implications for the health. And if a child is exposed to antibiotics, very young child or a newborn, and it disturbs the microbiome in the newborn, it has far-reaching implications for this child's future health. So all this knowledge came out, and now much more is coming out of this project. Before this project, the forerunner was Human Genome Project, which mapped our genes. The genes we inherited from our parents, our ancestors, and how they interact with the environment and produce disease, even cancer, metabolic disorders, diabetes, all those things. But the microbiome project now brought all that together. Microbiome and genetics are very closely interrelated. So much more knowledge is coming out. So microbiology connects all these things. It has many special branches, different microbes like viruses, fungi, bacteria. And then there are organisms which are neither bacteria nor fungi. Some have cell walls, some don't have cell walls. And now I must share this with you. An exciting thing about these microbes, the knowledge what we have so far, with all our intelligence today, the 21st century, we have finished quarter century. We are in the second quarter of 21st century. All that knowledge, we can put it as maybe 1%. There is 99% yet to learn for the human mind. And just an example, the permafrost. Permafrost is the frozen soil in the polar regions of the globe. It's now melting. And when it is melting, it's bringing out microbes which are millions of years old. Because our people are also digging them to find minerals, to find some precious metals or gems or oil, petrol. So when they dig it, they bring out those microbes, and then suddenly it is realized that the methane which is being produced, it's produced by these microbes. It's not so much pollution, dust and this and that. Yes, it contributes, definitely. I'm not advocating we should continue to, pollute the air, but a lot of methane, which is behind the ozone depletion, is coming from the microbes in the polar region. And those microbes may be zombies, some new pathogens may come out of it. So if you look at all those things, microbiology is the most fascinating science.
Yeah. Anyway, before I go any further, because you've also given us other more terms, maybe we could first start by, I'm going to take you to your first year class of microbiology, when the lecturer started by, what is microbiology? In the most simplest way, maybe we could first start by defining that, and then talk about, because we've also touched on pathogens. And another question, before this is the last one on that, so that you can just give us a whole, brief on definition and definitive of terms. So that is microbiology, and then you've mentioned we have fungi, we have virus, we have bacteria, we even have those that are neither. So if you could maybe give us a significant, how do we differentiate them? I know it's your job, but for the sake of the audience to get to have a beautiful view of what microbiology is.
I think microbiology, we are going back. We have already described microbiology, its scope already we have said.
Yes.
Microbes, I should say, are equivalent to God. God we cannot see, but we all believe in. God is everywhere, all around you, all over you, and inside you. You can describe microbes equivalent to God. And I think the most fascinating creation of God is microbes. And I think they are at the lowest rung of evolution. Evolutionarily, microbes are at the lowest rung, unicellular and even precellular. We have microbes which are described as prions. These prions are not bacteria. They don't have the structures. They're just a protein molecule. But it can cause disease. It can infect an animal or a human being. and cause very specific diseases and very difficult to find out. So from that, this goes to the primordial question, what is the origin of life? And they say that is the primordial secret. Nobody has been able to unravel. So you cannot define microbiology. You can simply say microbiology is the science of study of microorganisms which you cannot see with the naked eye. You need some aid to be able to see them. So you can simply say that it is study of microbes which cannot be seen with the naked eye. You need a microscope. Once it is under microscope, you can see them, they become microorganisms, and the study is microbiology. But we have already seen the scope, which is immense. It is not within this very narrow definition. So that's about it. But when it comes to the study, it's a highly specialized field. I have already described, yes. I gave you 10 branches. Yes, okay. There are another 30 branches of microbiology, because immunology, which is the science of defense, biological defense, an organism or an animal or a human being develops against attack of these microbes, is another science. It is also interconnected to microbiology. Now you come to subspecialties of microbiology. You have virology, you have bacteriology, you have mycobacteriology, which is the TB bacteria and the related bacteria, their study. You have mycology, which is the study of fungi. Now these fungi are another different class of most fascinating creatures. They are big, multicellular, they are unicellular, they are filamentous, they are yeast-like, the yeast forms, there are many, many forms, and they are all over in the environment. They are also normal flora of our gut. Okay. They also dictate our immunity. No. So the scope is extensive and very wide. There are many subspecialties, and the microbiology which I deal with, I should say, is called clinical microbiology or diagnostic microbiology. That is a defined area where we are actually diagnosing a particular disease in a patient. Like I deal with human health. If it is a vet, he is dealing with an animal. So you are diagnosing a particular microbial disease which is caused by a pathogen. Pathogen is the one which can cause disease.
Actually, that was my next question. So, we are talking about diseases, we're talking about infection, and this I learned that when we just talk about pathogen is disease
causing microbe.
Yes. But then we also have, now this awed me, and then when I kept on reading, I was like, now it makes sense. We have good bacteria, we have good fungi, but for virus, I don't think so. I'm not sure. I might be wrong. Yes. So there's good virus?
Your normal flora is composed of fungi, viruses, bacteria, all those other organisms, mycoplasmas, so the cell wall deficient organisms, all of them are there all over you, all inside you, like God.
Okay.
So the microbiome consists of virome and all other organisms. We call it virome, which is a normal viral flora of your body.
Okay, now that's something new I've learned. But then I wanted us to now separate that. When we just talk about the microbiomes, it's right. I got that right. That it's just not about it being infectious and it being, disease causative agent, which is now the pathogen. Talk about some that are good bacteria, good fungi, because this conversation is going to delve on the whole infectious, infectious pathogens or I think that will be me repeating myself when I say diseases. Exactly. So let's start with the good side. What does it mean to have good bacteria or good fungi? Where are they found? Before we get to now the...
See this, we touched upon this already. The microbiome, which consists of all this normal flora, which is in the animal body or human body, is protective. Actually, its function is physiological, protective, and also it dictates the immunity, the resistance to disease. So if you take an example of your mouth, you have normal flora in your mouth. There are many organisms. I should say for every one aerobic organism that which can grow in the presence of air, there are 990 anaerobic organisms, the organisms which you don't grow in the laboratory unless you have special conditions. And there are another 900 or 9,000 species of organisms which we have not described, we have not grown them, and we have not characterized them. There are many viruses, many bacteria in the nature we have not characterized, we do not know, we are not aware of. This holds good for animal kingdom also, animal pathogens, viruses. That is why I said the knowledge which we possess today could be just 1% or less than 1% of what is there to know. The knowledge is the ocean. What you have is just a pot of water you picked from that ocean. So whatever you have is this pot. The pot represents the ocean, but it is not the ocean. It cannot give you the dimension of the ocean. So that's how you can describe microbiology. There is yet so much to know. So the same organisms, some of them are also pathogens. They cause disease, like Staphylococcus aureus is a common bacterium you grow from pus. If you have a boil or an abscess or something like a small pimple, the tiny pus has Staphylococcus aureus. It is a pus-forming organism. We call it pyogenic organism. But it can be normal flora in your throat. There are many organisms which you carry, but they become pathogens. You carry many yeasts in your gut, but when you get sick, admitted to hospital, and you are seriously sick, the same organisms circulate in your blood and they are killers. And that is where the problem is. We call them opportunistic pathogens. They are waiting for an opportunity and they become pathogens. And what dictates this pathogenicity is displacement. If the candida is in my mouth, it's a normal flora, but if it enters bloodstream, it becomes something. But it can locally invade my mouth and then produce lesions. So there are many fascinating factors about pathogenicity of these bacteria. So my job involves diagnosing those conditions. It is a science which will not be defined and put into a capsule. Yes, people can do it if they have the skills to put it into 2 minutes capsules, but it's a study of minimum 5 years before we can practice. Before we are certified, minimum 5 years.
I'd like to understand now the training. What are you trained on? Aside from textbook, collective biology we had, because 5 years for me to even just get to understand maybe just one,
Let me tell you, there are many courses. You have a basic training, which is a microbiology MEd. It's a monospecialty program. You have 5 years, you train in microbiology. Whereas we have a program here, clinical pathology, where we have 4 subjects. One of them is microbiology. But we are teaching them at a monospecialty level because I'm a monospecialist. I set the lab and we are accredited by College of American Pathologists and the expectations are very high. So my postgraduates who are in clinical pathology program, which consists of many other branches, micro is one of them, they are at a very big level. But I trained 5 years in microbiology. One of my junior colleagues now in the department, who was my student who graduated, we sent her out for 2 years to train in microbiology. So now that 2 years microbiology will consist of a little bit of mycology, virology, bacteriology, immunology, diagnostics, quality control in microbiology, all those things. But all this revolves around 2 or 3 factors. The study of any microbe involves the structure of the microbe you study. This is part of the curriculum. Why do you want to study? The structure and the function, physiology. Why? There are 3 aims. Number 1, to design a diagnostic kit. When this pathogen infects an animal or human being, I know the structure, I am able to design a diagnostic kit. Number 2, to design a vaccine to prevent this infection. Number 3, to design a drug or another tool to kill this bacteria, to treat the patient. So there are three aims. One is to design a diagnosis, to design a vaccine, to design a treatment. Now to be able to do these three things, these are in any microbe study, these are the aims. To do this, you have to do so much research. Before I could detect one antigen or a virulence factor which causes the pathogenicity, makes this bug a pathogen, before I hit upon one, there is some 50 years of research which went on. Only thing now, research is faster because we have modern tools and the labs and things have changed. The world is going much faster. The velocity of discovery has increased. So we have many problems. We have also many solutions. We're continuously looking for solutions. But as we look for solutions, we control some microbes. We are getting novel pathogens. So there is a whole study within microbiology and infectious diseases of emerging pathogens, re-emerging pathogens. Emerging pathogens are new novel pathogens which are coming. Now we have recently, we have seen the headlines, Oropucho virus. We read about it long ago, we just forgot, we neglected. Now it's causing outbreaks. And now it will, maybe it's next pandemic, we don't know. It's just like our dengue, chikungunya virus, similar virus, joint pains, fever and incapacitating, mosquito borne. So new pathogens are taking upper hand. Then re-emerging is TB. We said we have controlled, notifications have fallen, but now suddenly it is re-emerging, more notifications, more sick patients are coming due to various reasons. But so emerging, re-emerging pathogens is a new branch of microbiology.
Now, the fact that you mentioned that a long time ago, some time back, you look into, maybe microbes or organisms, like, you even mentioned that a very new term, Oropucho, right? And then now we also have those that now, of course, like the dengue and the chikungunya fever, which can almost look or have almost like the same diagnostic map, if I got that right. And then also you've touched on TB, and it's even now coming up in more other ways, and you've used really scientific terms are the same in how they're evolving and even coming up. So what really entirely like makes an organism, become a pathogen or emerge? And if you could also define into it what makes it also re-emerge?
I think it's a very complex phenomenon. All organisms have some pathogenic potential. That is, they have some characteristics which can make them pathogenic. Some of them do not have that potential at all where they are, like some water organisms. They are normally present in the water, they are living, they are multiplying and they are going. We drink the water, they don't do anything. But there are organisms in the soil which are living there, but in the depth of the soil, if you dig and bring out that soil, and now this soil becomes dust, and then the human being is inhaling it. The human lung is not used to this organism which is in the depth of the earth, but you now brought it out. And it goes into the dust, and then the dust is inhaled, people can get sick. Most of the time we don't know what is happening until a scientist has gone, he looked into it, studied these patients, isolated that organism and has done the research to say that this is the pathogen. So Zika virus emerged like that. Sometimes there are many factors we know this is the factor. Climate change is a famous factor now for a lot of organisms to get displaced. Like in US, certain fungi were confined to certain states, but global warming has made them travel out of those states. They are now everywhere. Some fungi. Earlier, we studied that they are only confined to Arizona or New Mexico, this and that. But now they are found in New York, which is totally the opposite side. So what was in the East Coast is found in the West Coast, West to East, everywhere they are found. So Zika virus, when it emerged, we didn't know how it emerged as a mosquito-borne virus. We are given many reasons, but to date we don't know what exactly caused it. Global warming is one of them. Sometimes there are multiple factors. So, what they said, there was a genetically modified mosquito which was released in the South American countries. So, some scientists believe, and they have put forward some evidence, that these genetically modified mosquitoes are behind the emergence of Zika virus. Maybe it's the reason Oropucho virus is also emerging, and many other mosquito borne viruses are emerging. Now, when it comes to animal viruses, they are confined to some animals in the forest, in the depth of forest, where human beings are not exposed to them. But now deforestation, you cut the trees, and now these animals are thrown out. They are displaced, they don't know where to go. And the big trees are all cut, they are all felled. So they now come to the human dwellings. They are lost. And they say behind the emergence of a lot of arthritis, there is the displacement of deer from the forests in the US. So a lot of arthritis, which is a spirochete borne, the spiral organisms, which is, we call it Borrelia burgdorferi, is a famous organism. So the ticks, which are from the animals in the forest, the displaced, and then also when people go into the depth of the forest, they contract these things. And also we have these adventurers who went into the caves and then got
They pick something up
from the bats. And these bats are inside the cave. They don't disturb you, but you go and disturb them. So there is a lot of factors which are multiple. Global warming is one of them. Flooding is another of them. And this displacement of soil, permafrost, many things.
I'm going to try to be a good student with what I have understood in the most simplest way. So let's say this is an organism, okay? So with what you've defined, when it is in its natural habitat, it's harmless. If I take it out, bring it to maybe the first, in any way possible, maybe I went and picked it up somewhere, or maybe it was exposed to something else, like maybe a different unnatural habitat, it now can become a pathogen or can cause infections. The same way, here it can cause maybe something. Now this is a question. It can cause some different infection. If I take it out, or maybe I take it another one, this unequal to, bring it to another, it can cause another different infection.
This is not predictable, and it's not as simple as you have described. It's a very complex phenomenon. What makes it a pathogen has many dimensions. Environment is one of them. Its displacement is one of them, and which host it is. Like now when you displace the virus from the bats to many animals and human beings, all of them don't come down with the same disease. Some of them resist.
Yes.
Some of them don't resist.
I wanted that to come out.
Some of them become very seriously sick, like a hemorrhagic fever, where there is bleeding from all the body orifices.
Yes.
So this is dictated by the host. So this is another science, branch of science, which is called host pathogen interaction. And the third dimension is immunity. And the immunity is again dictated by the microbiome.
In fact, I think it's also a branch you also mentioned. Hey, science is broad. Exactly, science is super broad. Now, last question. Now, because you're talking about, for you to now, like you as scientists, for you to now come and understand what's going on here, there should be an emergence. You don't go digging yourself, because you used a very practical example, which I liked, where if, let's say, I dig up the soil, bring the organisms that are down there and expose them, and then maybe we breathe it through as dust, then something happens. Is that now when we start like now looking into it, or is just you scientists tend to now just also be ready? Are we in a state where we just are ready, or we wait?
That's a very good question.
Yes.
You see, you don't go and dig to look for a pathogen.
Yes.
It happens, you have patients, now you study those patients, and then you discover a new pathogen. However, the other way around is also possible. You have a patient, he has jaundice. This is how hepatitis C virus was discovered. He has jaundice. Now you take the blood, you take the tissues, and now you map the proteins. And from the proteins, they constructed the virus. It's not that they grew the virus, because there was no technique at that time. Now the scientists are looking actually for bacteria which have different characteristics, because we have immense commercial potential. We have bacteria in the ocean, Pseudomonas, which can actually synthesize many things we want to synthesize. And we, of course, all the antibiotics are from the soil microbes. Streptomycin, penicillin, they're all from the soil microbes. Many antibiotics.
You're making me come up with more answers. This is a really interesting conversation. So now that also means we can have one organism being a pathogen, and then we can use another one being a catalyst, or maybe being even a vaccine or the drug.
Actually, didn't answer what you asked. I was going there.
Oof, okay, I'm too fast.
So, you see, microbiome being protective and protecting the host through immunity, dictating the host immunity is one aspect. Supplying antibiotics, all antibiotics, including your penicillin, streptomycin, bacitracin, many antibiotics, everything ending with sin, was from soil microbes. They produce these antibiotics for their own protection, but we isolate them and we use them for our treatment. That is one aspect. The other aspect, now scientists are going out of the way and looking for new microbes because of the commercial potential. We can synthesize many things from the microbes. Even there is research to synthesize some petrol, synthesize proteins, many things which have commercial potential. So this permafrost, which I mentioned earlier, scientists went, and there is a whole research project funded by a consortium of Canada, America, and many other European countries, and it is digging soil in Antarctica, in the polar region, and then characterizing those microbes. And that is a very fascinating science which is going on. I must tell you, just 2-3 days ago, a finding has come out. There is an organism in the ocean which can switch off its metabolism and go into a suspended state for a long time. The bacterium is suspended. It is asleep. It's not multiplying, not dead. And it can stay there for any length of time, but can revive. So the metabolism is switched off, but it can be revived. So now these bacteria are found in absolutely clean rooms, where our NASA scientists, they are the ones who described this in Florida, the University of Florida, and they want these bacteria to be studied when they are taking a spacecraft which goes to the other planets. They want them to be absolutely free of microbes. But they found that this microbe can stay there and travel to the other planets because you cannot detect it with your normal tools. So you need some other tools to be able to detect these bacteria which are suspended. They are like in hibernation and they can wake up. So you might have many millions of such microbes in the frozen soil, in the depth of the soil, in the depth of the ocean, and this is the science. It's an ocean.
It makes sense with your intro that we are yet to know the 99%. We just have a bowl of water from a whole ocean. Oh my God.
So the fear is, when we go and dig the permafrost in the polar region, are we digging up some zombie bacteria which were pathogens maybe 2 million years ago and they are asleep? Now we bring them out. We don't have immunity. They can kill us very fast. They can wipe us out. We may or may not have time to design a vaccine, an antiviral, or an antibiotic, or even a diagnostic kit. It takes time to get these things. Though the velocity of this discovery is very fast now, especially with the advent of AI, artificial intelligence. These things are done very fast. I hope I have
This
sort
was,
of answered your
you've
question.
answered my question, and I'm learning a lot today. Thank you for that. And now to wrap this up, I'm going to have 2 questions, okay, for this session. And you're also going to have 2 questions for me. So my first question to you, if microbiology were a character in your life,
Microbiology was
a character, yes, in your life story, what would it personally be? Would it be curious? Would it be stubborn? Would it be full of surprises?
I think microbiology is God. God is everywhere. God is all around me. God is all inside me. And God dictates the life. Not just this planet, beyond the solar family, in the universe.
Okay.
And the secret of life.
So you would like an
And which can never be unraveled, which can never be mapped out completely.
Yes. Even when we
It's
try
immense, limitless.
That means in just one glass or even
word to
it has,
describe.
oh my God.
That word to describe, maybe all languages don't have that word. In classical language in India, Ananta. Ananta doesn't have a beginning, has no ending. It is Ananta. Microbiology is Ananta.
Okay, a question for me.
What made you choose me for this interview? I was wondering because I'm a very low profile person. I'm not a famous clinician or anything. For Daktari Online to choose me, what was behind this?
Okay, so it's to, amplify, just as from our intro, the health space practitioners, bringing all this together to have an understanding of things, in the health space. We just tend to see lab coats, surgery, but we don't get to have the story behind it. And of course, I was so excited to get to talk to, a specialist behind microbiology, because even you're surprised with the first thing I had a thought of, because these are the things we think out here and many others that I don't even maybe present today. And so when we get to have a great picture of who is behind this, what infectious diseases, the steps they have to, get to even get a vaccine or even a drug, and even the more research that has to go through that, because at the same time, again, you can get a drug that also even it's there, it's textbook, has side effects. Again, how do we deal with that? So it was really, I was excited because, just to get to have to understand the whole science behind it.
I still didn't get how you selected me.
No,
for so many things.
For selection, of course, it's guided. I wouldn't say that personally I would just be there and think of Professor Gunturu Revathi. We have inputs from various stakeholders and people in our office who we get to now suggest to have research. Exactly. And then when we do that, you, as much as I remember the conversation we had prior, I told you might see it as small for you, but for us it means a lot of things. From our side, is not how you see yourself. See? For us to see, exactly. So we see greatness, we see the science behind innovations and research, and infection is something, just as you said, it's likened to God. It's likened to something that has no beginning, and that means you're going to live in a world, and in fact, 1%, just from your own words.
Really, we don't know when microbiology began, because in modern school, medical school, we teach students microbiology began with Leeuwenhoek discovering the microscope. Not really. Look at the pyramids. The brains who constructed these pyramids, which are 5,000 years old, do you really believe they didn't know about microbes? And Egyptians who made the best of wines, the best of cheese, which are under 20 flavors of cheese, and preserved the dead bodies, which are even now perfect. They didn't know about microbiology. Microbiology, what is the beginning of microbiology, we don't know, and where it will end also we don't know. So microbiology for me is Ananta. Ananta is God. One of the names God is addressed in Hinduism is Ananta. No beginning, no ending. Microbiology is like that for me.
And that even now leads me to my second question. What is that non-scientific thing? Because you've given an example that maybe might be likened to the answer I expect, but I might be wrong. No expectation whatsoever. So what non-scientific thing has inspired your scientific thinking?
Non-scientific thing. I don't think anything can be called non-scientific, including arts. People say that fine arts are totally detached from science. It's not like that. That's not true. Art has an angle of science. Like if a beautiful painting, which is lasting, like Michelangelo has painted the Sistine Chapel, the murals he's painted. There was science behind it. He used colors, the chemicals, which were long lasting and beautiful. There is science. That science is required for Michelangelo as an artist to succeed. He needed to know that science to do those paintings. Similarly, a scientist needs to be an artist. He needs to know the artistic form of the science, discovery, innovation, new angle, look at it in a different angle, has an artistic angle. So for me, science and art are not different. Now, what is the non-scientific which inspired? Something like my grandfather, something like my father. My father is a poet. He's no more, but he was a poet and he was a thinker, and he inspired us in the daily life every minute to go and look for something different, look at things in a different angle, think about different things, and that's what inspires us to what we are.
Oof, great. A question for me.
Yes. Would you, as a journalist, find medical topics more interesting or literary topics more interesting to write about? A poem or a new book or a novel or a comic?
I find medical topics to be more interesting than literal. Okay, both of them have...
What reasons?
I'm going to give you my reasons. But then first, if I was to start by literal, of course, through my studying, when we were in class and the practicals and everything, we would first start with literal because we have not specialized. Just as in science and even in learning in school for doctors, nurses and all that, there's a whole collective class and then you start specializing. So for journalism is almost likely. So we'll do a collective class. So you're going to be bumpered with lots of topics. And then later in life, you'll have to decide that those who also still do multi-journalism, but then you can get to have a niche. And for me, if you can remember, I told you biology was never
So medical
exactly
branches so,
I just felt like it just called me. And every day it's interesting because
that's good. Wherever you are interested, yes, you go and read more.
Exactly.
And there's no limit these days. Information is explosion of information.
Yes, because today I will talk to you, tomorrow I'll talk to another specialist, and it's just mind-blowing because of it. It's just a learning day, and even sometimes you might think you know much, but then like
there's so much
exactly like for this session I'm just like, wow, okay, wow.
I think the knowledgeable one
exactly
is the one who knows that he doesn't know so much.
Yes, I do agree with that saying. And even I don't know, for some reason when you kept on talking, I was just thinking of Corona, the Corona period, and everything was not just like the pieces were making sense, like why we stayed in such a longer time, being asked to be at home because research was being done, and it's even now trying to even make me have an understanding of how that it was complex, and how it moved from different areas. And that's why even I understand why most countries will tell people to stay in there where they are at. And like you saying it, I was living it, I was reliving it, and I was like, whoa, okay, science is amazing. Science is beautiful. Yes. And so thank you so much for sharing this, and this comes to an end of our first segment.
Thank you too for this conversation. Yes. And I think I enjoyed it very, very much.
For our listeners, that is Professor Gunturu, who has taken us into the depths of the microbiology world and her life towards the path of being a microbiologist and a specialist in infectious disease. For our next segment, we're going to touch on things that happen in our households, that is antimicrobial resistance. See you there.